Okay. Okay. Good evening. Good evening. Okay. Go to the Theut. We discussed up to bioremediation last week. No, up to bioremediation completed. Today we going to start. I told the bioremediation mainly two type: natural bioremediation and artificial bioremediation. Do you remember what's the meaning of the bioremediation? What's the how we define the bioremediation? Quickly, anyone? How we define the bioremediation? Biological remediation. Bioremediation, how we define? Quickly. Yes. Environment hazardous compound removing or decrease the hazardousness, hazard the value by whatever organism. That's the meaning of the bioremediation. The bioremediation mainly two types: natural bioremediation and artificial bioremediation. Natural bioremediation is use environment existing microorganism or whatever organism for the bioremediation process that we called as the natural bioremediation. The artificial bioremediation is in laboratory cultured microorganism or organism introduced for whatever environment to reduce the hazardness of the environment. Okay, that's we called as a bioremediation. Bioremediation two types: natural bioremediation and artificial bioremediation. Okay, completed. Okay, you can remember a few examples. I told in artificial bioremediation introduce different microorganisms to remove oil sprints in oceans and remove micropolymers, microplastic in oceans and aquatic environments, remove microplastic, and different purposes we use this artificial bioremediation. Artificial bioremediation. Okay, final one: microorganism used in wastewater treatment plant. Okay, in wastewater treatment plant, a lot of MCQ questions coming from this part: wastewater treatment. Actually, the wastewater today we going to discuss wastewater treatment. The wastewater treatment is different from the drinking water treatment. The drinking water treatment, the same process, but more than advanced. The drinking water treatment, it is being by our water board. It is little uh, advanced part. But today we going to discuss this wastewater treatment also very important. Today we going to discuss how we use microorganism for the wastewater treatment plant. Okay, in wastewater treatment, there are three main steps. Wastewater treatment, there are three. Sometimes they called sometimes it is given as 4 steps, but in our syllabus it mentioned actually it has 4 steps, but in our syllabus it has given as 3 steps. Okay, wastewater treatment. Wastewater treatment. Wastewater treatment mainly 3 steps. The first one is the primary treatment. Primary treatment. The second one, easy to remember, secondary treatment. Secondary treatment. And final one is tertiary treatment. Tertiary treatment. Tertiary treatments. Okay, they are the wastewater treatment steps. Okay, primary treatment. actual wastewater treatment plant, there is additional treatment process that we call as preliminary treatment. මේකට තව එකක් add වෙනවා අපේ syllabus එකේ නෑ, that we called as preliminary treatment. Preliminary treatment. Okay, in exam, if they ask what are the 4 main steps of wastewater treatment plant, you can write like this. But in our syllabus, it is given as dividing into three steps: primary treatment, secondary treatment, and tertiary treatment. Okay, primary treatment, secondary treatment, and tertiary treatment. Okay, now you know the wastewater treatment is not only for single industry. Normally wastewater treatment plant is working for several industries, normally industrial zones. It can industry not for a single industry, it is a industrial zone. Okay, then different kind of water collect into the wastewater treatment plant. Okay, that we called as industrial zone. I think in chemistry, yesterday chemistry, no, last week chemistry part question, we discussed about industrial zone. In industrial zone, huge volume of wastewater is generated in different production industrial process. Okay, then think different cement industries, rubber industries, and cement, rubber, and different kind of chemical industries, different industries in a same area that we called as industrial zone. Industrial zone. Okay, for this industrial zone, there is a water treatment plant. It is compulsory to maintain water treatment plant to treat the industrially used water and release to the environment. That's the purpose of wastewater treatment plant. Treat the industrially used water. Industrially using mean, you know, there are lot of hazardous compound, hazardous microorganism, chemicals add with water. Then if it is directly exposed to natural environment, it is a huge damage to environment, into aquatic ecosystem and the total environment. It is a huge damage. Therefore, we can minimize this, only minimize. We cannot 100% treat this by this type treatment. We can minimize this damage by using wastewater treatment plant. Okay, wastewater treatment plant. Now we're going to discuss about wastewater treatment plant. This is very important lesson. Therefore, don't miss any part. Okay. Wastewater treatment plant, we can divide into 3 parts, as I told: primary, secondary, tertiary, and finally release to the environment. Okay. Now, the first, from this, every industry, this type wastewater treatment plant we called as central wastewater treatment plant, because from every industries the wastewater collect into one treatment plant. Okay, therefore that we called as central wastewater treatment plant. Different types of wastewater treatment plant, but their wastewater treatment process is similar, same. Okay, now first of all, this collected water, you know, collected water, different compositions, different chemical levels, different temperatures. I got. Then in different industries, different composition, different concentrations, different chemicals, different temperature level water is collected to this treatment plant. Actually, it is not favorable for treatment process. Therefore, actually this preliminary treatment එකෙන් කරන්නේ මේක අපේ එකේ නැති වුණාට, the preliminary treatment, it is make as equal composition, equal concentration solution එකක් කරන එක. Understood? Preliminary treatment mean the collecting water from different industries. It is actually uniform concentration, uniform composition, uniform temperature, mixture, water mixture. The the meaning of the preliminary treatment. I got the preliminary treatment. Mainly it is this process, it is turning, water is turning and mixing, it is converted into equal composition of their composition. Understood the preliminary treatment? Preliminary? I need answers. Yeah, you have response because this part is not it is new for all physical students. Same, it is new. Okay, therefore focus real. Again, I am not going to do this lesson in physical classes. Okay, now physical classes cell structure is completed. Cell structure, only this part is remain. Okay, this part you can cover from this class. Okay, that's why it is compulsory and important. A lot of MCQ and small questions are coming from this part. No more marks, but we have to cover. Okay. Okay, the preliminary treatment, the different composition water types come into the wastewater treatment plant. In this preliminary treatment, what we mixing and make a equal composition solution. Equal composition solution. That's the preliminary treatment. Okay, preliminary treatment is complete. Now, what is the meaning of primary treatment? In primary treatment, it is actually physical process. It's a physical process. MCQ questions, it is asked. It is a physical process. Physical process. In primary treatment, it is the first in wastewater treatment, we can't treat wastewater with solid particles. Large solid particles, we can't treat any water. Therefore, in first step of the primary treatment, in first step of the primary treatment, the first one is the filtering. Different type of filters we can use, bar screens and rotary screens. Different types of screens we use to filtering process. Okay, filtering. Okay, then filtering and separate solid particles and large solid particles. The suspended particles we cannot remove by filtering, but large solid particle, floating large, as example, think cloth parts, the plant leaves, like large solid particles we removing by filtering, by using screens, different types of screens, bar screens, rotary screens, like this. I will show you what are the types of screen and how it is doing. Okay, after that, this filtered water transfer into a large tank and keep some time for settling out means suspended particles. The main target is suspended non-dissolving suspended particles. We, in primary treatment, suspended particles we need to keep some 24 to 48 hours. Then what happened, the suspended particles is go down, precipitate. Okay, that's only primary treatment. First, filtering solid particles and separate solid and water. Then water is transferred to settling tanks that we call as settling tanks. settling tanks, then keep some time for settling out. Sometimes we can add aluminium sulfate that we called as aluminium sulfate, aluminium sulfate. Aluminium sulfate. Aluminium sulfate is alum. We can add, then the alum is coagulate. Coagulate means these suspended particles are combined, then large particles produce. That the precipitation is easy. Okay, if it is sometimes adding alum, sometimes not adding that, but this is totally physical process. Okay, I got the primary treatment. Very simple, very easy. I got the primary treatment. Any doubt in primary treatment? In primary treatment, first remove solid particles and separate water and transfer to a settling tank. In a settling tank, non-dissolving suspended particles, precipitation is the keep some time for precipitation. That's the target of primary treatment. Understood? Quickly. Amar, I got. Please send me yes or no, then it is easy to. Shehan? Sadeep? Okay, this is a primary treatment. In primary treatment, first separate solid particles from the wastewater and the filtered water transfer to settling tanks. In a settling tank, we can add alum to increase the speed up this process. The precipitation process we can speed up by adding alum. The alum is a chemical, but commonly we consider the primary treatment is a physical treatment process. Okay, second one. Then after that, this water, after that, we remove this suspended precipitated layer. We can separate, and this water we can separate the above water layer without suspended particles. We can transfer to the secondary tanks. Okay, it is transfer to secondary tanks. Secondary tanks. Now the secondary tank I will drawing here. In secondary tanks, give me a second. In secondary tanks, the secondary treatment process is a biological treatment. Remember, it is a biological treatment process. The secondary treatment process is a biological treatment process. It is a biological. The first one, the primary treatment is a physical treatment process. The secondary treatment process is a biological treatment process. In biological treatment process, what happened? We need to introduce some microorganism, mainly bacteria, specific bacteria type. For the target is in these secondary tanks. The target is, in primary treatment, suspended non-dissolved particles, paven and suspended, floating and suspended particles removed by primary treatment by settling out. In secondary treatment, our target is these non-dissolved particles. Now if it is if we remove non-dissolved suspended particles, it is already available in this tank, in secondary tank, dissolved particle. Our target of this secondary treatment, remove these dissolved particles. Dissolved particles, our target is these dissolved particles, dissolved suspended solid, that we call as DSS. DSS mean dissolved solid. DSS mean suspended solid. Suspended solid. Okay, dissolved solid, we need to convert into non-dissolved solid by using bacteria. Special type of bacteria activity. This dissolved solid, we need to convert into non-dissolved solid. Then after this conversion, the dissolved solid convert into non-dissolved solid. For this process, we need bacterial activity, bacteria or microorganism activity. They compulsory to introduce aerobic microorganism. For the wastewater treatment, actually mainly two types: aerobic wastewater treatment and anaerobic wastewater treatment. But in our syllabus, we mainly discussing about aerobic microorganism. Using aerobic microorganism, the reason is their activity, this dissolved compound convert into non-dissolved compound speed is very high comparatively anaerobic treatment. Actually, anaerobic treatment we cannot do in small scale. It is a lot of maintenance and the initial cost, because we need a full cover treatment process, sealed treatment process. Because if there are any if there are any oxygen particle enter, what happens? It is toxic for microorganism. Therefore, we mainly discussing about aerobic microorganism, aerobic treatment process. For aerobic treatment process, then what we need to do? What you need to give? We need to give oxygen. We need to give oxygen. Okay, now for this secondary treatment plant, okay, again come to this primary treatment. I will explain from the beginning. First of all, in preliminary treatment, what happened? The all types of wastewater convert into equal composition solution. After that, screening by filtering, filtering and solid particles removing. Then this filtered water fill into settling tanks and settle down this suspended solid, undissolved solid, and after that separate this non-dissolved solid and transfer into biological treatment plant, biological secondary treatment plant. In secondary treatment plant, we need to introduce a special type of microorganism. Bacteria introduced. For while react with this microorganism, what happened in water? What happened to the DO value of water? Dissolved oxygen level inside water. This bacteria, all bacteria working on this water, they are aerobic microorganism, aerobic bacteria. Then what happened to the DO value, the dissolved oxygen value of the water? It is increasing or decreasing? Dissolved oxygen value. Okay, what do you think? When we introduce aerobic microorganism into these tanks, when they react on these organic materials, dissolved compounds, what happened to the DO value, dissolved oxygen level? What happened? Denith, what happened to the dissolved oxygen level in the water? Quickly, quickly. We have a lot of parts to discuss today. What happened? Melfa, what happened? What happened to the DO value? DO mean dissolved oxygen value. The oxygen concentration of the water increasing or decreasing? Simple question. Oxygen concentration increasing or decreasing? No idea? Simira? Amavi? Simple. I am asking, when we introduce aerobic bacteria into secondary tank, and when they act on dissolved dissolved organic compound in the water, then what happened to the water DO value, dissolved oxygen concentration? Very good. It is decreasing. Dissolved oxygen concentration is decreasing. And what happened to the DO value? Sorry, DO value is decreasing. What happened to the BOD value? BOD එකට මොකද වෙන්නේ? The BOD, biological oxygen demand. Biological oxygen demand increasing. Very good. The biological oxygen demand increasing. Therefore, actually when decrease the dissolved oxygen, automatically BOD and COD values increasing. BOD, COD values increasing. Okay, the problem is this bacteria use water available, water dissolved oxygen, and decrease the oxygen level in the water. Decrease the oxygen level in the water. Okay, for when they are growing and reproductive, and for their development, therefore oxygen deficiency is coming. Oxygen amount is decreasing. Therefore, we need to artificially provide oxygen for this tank. We need to provide oxygen artificially. Artificially, we have to provide oxygen. Okay, these oxygen supplying methods, there are different types of oxygen supplying method. The first one is we can bubbling oxygen by using pipes and we can provide oxygen artificially by using pipes by water bubbling, not oxygen bubbling, like a fish tank. Like a fish tank, we can provide oxygen by tubes. And we can again we can use, the first one is oxygen bubbling, oxygen providing methods. Oxygen supply methods. The first one is oxygen we can provide artificially by oxygen bubbling. Oxygen bubbling. Like a fish tank. Okay, in fish tank we use a motor and we provide oxygen. samely we can provide oxygen for this tank. It's a large tank. It is not easy like fish tank. It is difficult process, but we can. Okay. The second one, we can use trickling tank, trickling method, trickling tank, trickling tank, trickling tank, trickling tank method, trickling tank method. Every method you have remember. Trickling tank methods. Always in structured paper asking trickling tank method. I will show you what is the shape and how it is aeration method. It's also aeration method. Okay, that we called as trickling tank method. The final one is aeration barrel, rotating barrel method. Rotating barrel method. Okay, they are the aeration methods. Okay, I think you now it is clear. It is very simple process. After filtering, it is settling out in primary treatment. Then non-dissolved particles, suspended particles are precipitation. Then we can separate. Okay, after that, the only this separated this water contain only dissolved particle. By using microbial activity, we dissolve particle convert into non-dissolved particles. Okay, then when we introduce these microorganisms, they are aerobic microorganisms. Therefore, the water oxygen level is decreasing, the VOD level is increasing. To reduce that, we need to provide artificial oxygen supply. For artificial oxygen supply, we have three methods: oxygen bubbling, trickling tank method, and rotating drum or rotating barrel method. Rotating drum, rotating drum method. Rotating drum method. Okay, are you clear up to now? Are you clear up to now in treatment process? Okay, I need responses, otherwise how I know? Please, if you any doubt, please ask, and I can explain easy. I got up to now? Okay, now I will explain this oxygen supplying method. For oxygen supplying method, we already know the oxygen bubbling is very simple. Oxygen bubbling means using pumps, we can provide high pressure oxygen into water. That is a simple method. This trickling method is important. Trickling methods. For trickling method, we need special type of tank that we call as trickling tank. Okay, trickling tank. Trickling tank, it is like this. The trickling tank is like this. Understand, okay, this is a very important process. Okay, that we call as trickling tank. Trickling tank. It's a large tank with filled with stone. Filled with stone. I'll show you in videos. Make it stones gal purawala tama තියෙන්නේ. Filled with stone. Filled with stone. Sometimes some industries use plastic pellets. Plastic pellets. Sometimes we can use stones. Stone. Okay, the special type of microorganism. Special type of microorganism growing on these stones or plastic pellets. Then the primary treated water spray out onto this stones. That is spraying. Spray. Okay, spray into these stones. then these special microorganism growing on the stones, growing on the stones, then this water, when it is come through in the from the atmosphere, it is aeration. Atmosphere, lot of 20% oxygen, therefore it is aerated, and when it is go through this go between these stones, then what happens? It is react with microorganism. The microorganism doing their job, then it is convert dissolved solids convert into non-dissolved solid. Dissolved solid convert into non-dissolved solid, that we called as trickling tank method. Trickling tank method. It is an aeration process. Aeration process. Are you got? Amma, we got? Trickling tank method. Amar? Okay, I will show you this every steps. In trickling tank method, microorganism, the special bacteria is developed. They growing on the surface of the stones. Surface of the stone. Okay, the final steps, the final aeration method, it is rotating drum method. The rotating drum method also same. We need large tank, and it is connected with this type large barrel. This is a drum, rotating drum. Okay, it is the special bacteria is growing on the surface of the drum, and it is rotating. It is always slowly rotating. Now you can see some part of the drum is touched with the water. Then the microorganism act doing their job, and when it is go up, it is aerated. Microorganism aerated. Then they can grow very well on this surface. They can grow and they can doing their job. But this method, the efficiency is low than trickling method. Trickling tank method is efficiency is high due to because always microorganism contact. Always microorganism contact. When this water come through out the when it is precipitate, again it is circulate process. It is circulate process. Therefore, again and again, same water amount contact with microorganism. Therefore, the trickling tank method is more efficient. Okay, the other method is the oxygen bubbling method. Okay, that's the secondary treatment process. In secondary treatment process, what is the purpose? Dissolved suspended solid convert into non-dissolved suspended solid. After that, after this bacterial process, again this water transfer to settling tank, again transfer to settling tank, and keep some time. Then suspended particles go down. Then we can separate. Then when complete the secondary treatment, in the primary treatment we completely removed suspended non-dissolved solid. In secondary treatment we can remove dissolved solid in the water. Then total suspended and dissolved solid is removing at least 95 to 96% we can remove. Understood? That is a secondary treatment process. Okay, any doubt in secondary treatment? You can ask. Any doubt in secondary treatment? If anything you want, you can note down, because some parts is not given in the tute. Some part not given in the tute. If you need a special thing, you can note down. Okay, any doubt? Please quickly. Any doubt? Any doubt? No. Okay. This is primary treatment. What we did in primary treatment, non-dissolved suspended suspended solid. We can remove in the primary treatment. I told it is a physical treatment process. Physical treatment process. In the secondary treatment, we remove dissolved organic matters, dissolved suspended solid. Then that we called as biological process by using bacterial activities, microorganism activity. Then finally treatment. Finally, it is tertiary treatment. The tertiary treatment process that we called as a tertiary treatment process we called as a chemical treatment process. Chemical treatment process. Okay, after primary and complete the primary and secondary treatment process, this secondary treated water we transfer for chemical treatments. In the chemical treatment, there are different chemical treatment process. Okay, there are different chemical treatment process. In Sri Lankan condition, the main method is chlorination. Chlorination. The chlorination mean you already know the chlorination mean it is add chlorine, adding chlorine. The second, there is another method that not used in commonly in Sri Lanka. It is ozonation. Ozonation. They are the main chemical treatment process, tertiary treatment process. Now you can understand after a secondary treatment process. After secondary treatment process, it is after settling out this separated water. After that, it don't have any suspended solid or dissolved solid. Then finally, it is transfer for chemical treatment process. Chemical we add ozone O. We react with ozone O chlorine. That simple process. Then after that, it is exposed to the environment. Okay, that's a full production treatment process of wastewater treatment. Okay, any doubt? Final thing. What is the purpose of this tertiary treatment? What is the chemical treatment purpose? In primary treatment, remove suspended solid. In secondary treatment, remove dissolved suspended solid. In tertiary treatment, what is the target? What is the purpose? Any idea? Why we doing tertiary treatment process? Without now, if there are no any solid, it is 90%, like approximate 90% it is treated. Why we not directly? It is a very expensive process because we add chemicals for water. It is expensive process. Why we doing this? More than very good. More than primary and secondary treatment, this is an expensive process. Okay, without directly removing water to the environment, why are we doing this one? Anyone? Yes, some are correct. Others? Others? What's the purpose of doing this? I need the answers. Yeah, I think you already know it. What's the purpose of doing tertiary treatment? Only two answers. Others? Quickly, quickly. Yes, correct. Yeah, these two answers correct. Remove harmful microorganisms. Yes, if the treated primary and secondary treated water, in secondary treatment we doing a bacterial process, no microorganism reacting process. Therefore, it can it can contain some harmful microorganism to environment. Okay, like salmonella mainly. Salmonella, 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 and E. coli. E. coli is Escherichia coli. Escherichia coli. Salmonella and salmonella species and E. coli species, if it is exposed to environment, it is a health effect for humans and other animals. It is actually it is harmful for environment. Therefore, we need to remove by these chemicals. Adding these chlorine-like chemicals, we can decrease their concentration. We cannot fully destroy, but we can control. We can control and get into environmental favorable concentration. Environment favorable concentration. Then after then we can check the parameters, and finally we can remove to release to the environment. Release to the environment. Okay, when it is released to environment, okay, I will show you the video and let's come to the parameters. Okay, if you treat it in wastewater treatment plant, you cannot satisfied and you cannot satisfied in your level and you cannot release to environment. There are some guidelines, there are some standards. Central Environment Authority, CEO, monthly or weekly, they come to the treatment plant and they take samples and check these parameters. And they recommend some treatment process. Okay, therefore it is a legal process. Actually, it's a legal process. We need to maintain some standard after treatment when it is released to the environment. Okay, I will explain later this is standard. Okay, this is a video. Oh, wait, I think that's there.
The Spanish Fork wastewater treatment plant has been.
Right. Oh, give me a second. That's right.
Spanish Fork wastewater treatment plant has been operating since 1956, over 60 years with major upgrades.
Can you hear sound? Is it audible? Not now, previous. When I play in this?
In the mid-80s. During this time, the amount of wastewater processed has grown between 4 and 5 million gallons every day. And with an ever-increasing population, it is no wonder Spanish Fork City is beginning the process of building a new wastewater treatment plant. So sit back, relax, and join us on a tour of the current plant as we describe how it operates, emphasize the need for a new treatment plant, and help you understand how we each can make our sewer system stay clean and functional. The treatment process begins when wastewater for a new treatment plant and help you.
The industrial zones. It is collecting collecting water from all industries into one place. That we call as central wastewater treatment plant. Okay.
Understand how we each can make our sewer system stay clean and functional. The treatment process begins when wastewater, which is anything that runs down your pipes, arrives at the treatment plant and enters this building. The wastewater passes through step screens specifically designed to remove large solids.
They are the different types of screens. Am I audible? Am I audible? Audible. Okay, there are different types of screens. You can see here, they are bar screens. You can see solid particles separating. Only water is filtered into the treatment tanks. They are the solid particles, shopping bag, okay, coming with wastewater.
Solids which should never be in the sewer system. Please do not flush diapers, so-called flushable wipes, feminine hygiene products, condoms, makeup kit accessories, clothing, or any other inappropriate material. These items can cause unpleasant.
They are removing parts
and costly repairs to pipes within the water collection system. Anything that is caught in the step screens is removed, washed, and taken away to the landfill. Whatever passes through the step screens comes here to the grit chamber. This chamber is designed to allow heavier solids such as dirt, sand, and rocks to sink to the bottom. This allows only the wastewater to flow through to the next stages of the treatment process. The velocity of the water is slowed and air is pumped through the pipes into the water to facilitate this separation. The solids are pumped off the bottom of the grit chamber and also taken away to the landfill. The waste removed from the step screens and grit chamber combined is about 1 ton per week. We again strongly urge you to only flush appropriate disposable material. The wastewater is now finished in the grit chamber and is split into opposite directions, leading to one of our 3 primary clarifiers. Primary clarifiers use the law of gravity to allow the dense material.
They are preliminary treatment. Okay, the previous part is grit chamber is equalization process. It is mixing and all water, and it is flowing with shallow area. It is a equalization process. After that, it's come to, after screening, it's come to primary treatment. Okay, primary treatment.
Material called sludge is settled to the bottom of the large basins. The water comes up through the middle and slowly makes its way toward the outside wall, spending an average of 5 to 6 hours in the basin, all the while allowing the solids to settle to the bottom. There are sweeping arms on the top and bottom of the primary clarifiers, used to either get rid of floating debris or to collect the sinking sludge, all of which gets pumped to the digester. Much of the foam and other icky debris you see on top.
Okay, they are the primary tanks. Primary tanks mean they are clarifiers that settling out tanks.
Okay, the water is supposed to be previously removed in the grit chamber. This is yet another reason
They are the
new treatment
suspended
plant is being built
parts.
to replace old or damaged equipment
Suspended
and
parts.
outdated processes. 90 to 95% of the settlable solids are removed by the time the wastewater leaves the primary clarifier, and 40 to 60% of the suspended solids are removed as well. The water now flows from the 3 primary clarifiers to this building, which is known as the intermediate pump station. The wastewater flows in and is brought to a higher tank, all accomplished with a screw pump. Each screw pump is capable of pumping as much as 10 million gallons of water every day. From there, most of the wastewater flows to the aeration basins, while some flows to the trickling filter.
In our discussion, this is the secondary treatment. Okay, focus well. Now, first we discussed in the video. First they did preliminary treatments, filtering and equalization. They are preliminary things before primary treatments. Then primary treatments, they doing the settling process. After that, they directly transfer to the trickling tank. Trickling tank is trickling tanks.
On top of the trickling filter, you can see the wastewater is sprinkled over this plastic media.
They not use stones. They're using plastic pellets.
So the vast and multiple layers of media is to expose the wastewater to a large surface area of good bacteria growing on the plastic. This removes any bad bacteria as well as harmful chemicals such as ammonia. Although the trickling filter used to be a cost-effective and natural form of wastewater treatment, it now no longer provides the quality of cleanliness needed to comply with current standards, which includes nutrient removal, a process the new treatment plant will better accomplish. The water that does not find its way to the trickling filter flows to one of these 4 aeration basins. In each basin, we find good bacteria or microorganisms to help treat the wastewater. The large wheels.
Okay, that I explained rotating drum method. They're using different names, but you have to remember this is a drums. They are drums. Sometimes some completely roller drums using. Now to increase the surface area, they are using different shapes. Okay. Okay, that's a drum method.
Waste from the sewage provides the food. Since air is provided, this is an aerobic treatment process. It's a natural process harnessed and expanded to fit the city's growing demand and provides all that's required to keep those cute microorganisms happy in a good growing environment. After flowing through either the trickling filter or the aeration basins, the wastewater comes here to one of 2 secondary clarifiers. Much like the primary clarifiers we saw earlier, these
secondary clarifiers, after dissolved solid in bacterial process, after that they convert into suspended solid. Then in the suspended solid, after completing this bacterial process, it convert to secondary clarifier. Secondary clarifier mean again settling out process. Okay, settling out.
These clarifiers allow the denser material to sink to the bottom. A large portion of the activated sludge that collects on the bottom of these clarifiers is returned to the aeration basins in order to reuse the microorganisms, while any excess is pumped to the digester for further treatment. After spending approximately 18 to 24 hours in the treatment plant, the wastewater now flows through the chlorine contact basin. This chlorinator pump takes the chlorine from its holding tank and mixes it into the water. The chlorine contact basin slows the water flow through these long channels and gives the chlorine time to perform proper disinfection before discharging the clean water into Dry Creek.
Then after
Wait a
chlorine
minute.
process,
We forgot about the solids and sludge from earlier. Let's go over to the digesters. Unlike the aeration basins that use an aerobic treatment process, the digester operates on anaerobic treatment, or treatment without air. Through technology and innovation, we again have adapted the natural biological process similar to what happens inside a human digestive system and produced it on a very, very large scale. It takes a minimum of 15 days for the sludge and solids to pass through all 4 digesters and move.
After that, it is sludge processing system that you in our syllabus that not mentioned, but you can put this remain part. It's important. Now the water is released to the environment. The collected sludge is processing that we call as sludge processing.
Move on to the next step of their treatment process. As the sludge comes out of the digesters, it still has a high liquid content. So in order to remove some of that liquid, it comes here to the belt press. As the sludge is pressed through these folding layers of the belt press, it goes from 4% solid to almost 18% solid. Now that may not seem like much, but remember.
Okay, the belt process, the belt pressing is the sludge. First of all, we remove the sludge and water. The water concentration is decreasing, then you can see it is now it's like a solid matters. Now it is pressing well and drying process. That's the main target of this sludge processing. Water we need to dry this sludge. Dry. What's the purpose? Oh, do you know? I'll explain after that.
Your body is 40% solid matter. The sludge makes its way out of the belt press and is collected here. This treatment process is so thorough that even flies, cockroaches, rats, or other rodents are not attracted to this newly dried material, which will either be used as fertilizer on farms or taken to the landfill. As we look to the future, the new wastewater treatment plant will be critical in meeting regulations of cleanliness and safety for Spanish Fork City. Now that you have learned what not to flush, as well as the time and effort that goes into treating wastewater, we hope you will be more cognizant of the role each and every one of us plays in making sure our wastewater system flows smoothly.
Okay, understood? Any doubt now? Okay, do you know what they told in video? What is the final outcome for sludge? What they doing finally for sludge? It's not a Sri Lankan one. What they doing? Finally, finally they told after drying this sludge, reducing water amount after drying, compost and landfilling. But sometimes some industries it is dangerous. In Sri Lankan condition, we not use this sludge for landfilling because it contains some heavy metals. Do you know in Sri Lankan condition what doing for sludge? Normally, you know what's the company? Cement company. What's the name? I can't remember. In cement production, for calcium carbonate digestion, It need more than 1800 Celsius. No, for this digestion process, we can use this sludge to generate the heat. Generate the heat, especially this textile industry wastewater sludge we use for cement productions to generate the heat inside the boilers. Okay, they are the wastewater treatment plant. In the wastewater treatment plant, when it is finally, we're not going to discuss about sludge in your syllabus, we only discussing about water, wastewater. Then I told when we release to the environment, lot of parameters we need to maintain. We need to keep the parameters in standard range. Okay, now these parameters we can divide into 3 parts, as I discussed again yesterday. The parameters, standard parameters, mainly 3 types. It is physical parameters, chemical parameters, and biological parameters. Okay, they are not given in the tube, you can note down. Okay, physical, biological, and chemical. Okay, physical parameters are color of the water, physical, temperature, physical, turbidity, physical. Turbidity means the amount of suspended solid. Turbidity. And there is another one, TDSN, total dissolved solid amount, and TSS, total suspended solid. Total dissolved solid and total suspended solid amount. TDS and TSS. They are the physical quantities, physical parameters. There are other types of parameters, chemical parameters. Remember pH value, BOD, biological oxygen demand, DOD, dissolved oxygen level, chemical oxygen demand, COD, and what else? Acidity, and all are dependent on pH value, and each and every heavy metal concentration. Heavy metal concentration also in chemical treatment, chemical parameter. Arsenic, phosphate, what else? Heavy metals, mercury. The concentration should within the approvable range. Okay, biological parameters, we can measure the salmonella and species count, and E. coli, Escherichia coli. Okay, that's the full wastewater treatment part of the cell structure. Okay, if you have any doubt in wastewater treatment, please ask. Okay, go to page number 30. Page number 30, you can fill this space, preliminary treatment, primary treatment, secondary treatment, but in your syllabus, it's not given preliminary, but no problem, you can. Preliminary treatment, primary treatment, secondary treatment, and tertiary treatment. Write down the preliminary mean the equalization process, equalization process. The primary is a physical, secondary is a biological, tertiary is a chemical process. Quickly note down. Okay, any doubt in wastewater treatment? Everything clear? Send message. Everything clear or any doubt in wastewater treatment? If you go to the tute, I have given the same process I have given as a chart. Okay, this one. Wastewater inlet and the buy screen. After that, settling tank, sedimentation tank, aeration tank. This is a primary treatment, secondary treatment, finally tertiary treatment. And this is sludge processing unit. Okay. Okay, final one. Use microbes in biogas production. Final production process. Okay. Biogas production unit. I'll check your MCQ marks our every answers and mark schemes I upload to group. You can check your MCQ marks. When finalize marks, I will check if is it correct or not. Now you can check and fill in the given sheet. Okay, now I think only 8 or 9 student only filled. Others please filled it. Finally, after these 3 months, when you go to exam, you can find your line and you can find your performance, how you work, and you can improve one by exams. Okay, by this method. Okay, please fill the sheet. Only MCQ marks you need to enter. This one only 9 is filled. Yeah, fill this your name and marks, MCQ marks. Okay, give me another one. Wait, in waste steps in wastewater treatments, according to your syllabus, it should be like this. Preliminary treatment is not compulsory. Okay, the primary, your answer should be primary treatment, secondary treatment, tertiary treatment, and final one, anaerobic sludge digestion. Anaerobic sludge digestion. Okay, they are the main steps. If exam asks what are the main 4 steps in wastewater treatment plant, you need to explain these 4 methods. Preliminary treatment, you can, if you want, you can add to primary treatment. Okay, they are the main steps. Okay, good. Okay. Now, note down. Okay. They are the main 4 steps. The final one should be anaerobic sludge digestion. Anaerobic sludge digestion. Okay. Okay. Now we move into our microorganism used in biogas production. I told earlier the biogas production is a fully anaerobic process. Fully anaerobic process that therefore you already know it must have a fully closed environment, fully closed chamber actually. Okay, this is a biogas production unit. This is a biogas production unit. There are different types of biogas production units. That's a common type. Biogas production unit. This is the inlet. The inlet mean raw material inlet. Raw material inlet. What are the raw material we can use for biogas production? Any idea? Let's say effluent mean raw material input tank. We can input raw material. What are the raw materials? Raw material. What are the raw materials we can use for biogas production? Any idea? Raw materials we can use for yes, kitchen waste is a good source for biogas production. And animal waste, cow dung, like animal waste is a good source for biogas production. What else? Garden waste. Garden waste. What do you think? Garden waste is good substrate or no? Any idea? Garden waste, garden waste mean the dry leaves, and if we consider straw වගේ එකක්, පිදුරු, straw like material, is it okay for biogas production? It is okay, but we have to maintain same as compost production. The biogas production also doing by microorganism, special type of microorganism. Therefore they are favorable substrate, they are favorable conditions we need to maintain inside this production process. Therefore for biogas production we need to provide favorable production process, favorable environment for their microorganism growth. Biogas production. We need to provide favorable environment. This favorable environment is normally they need the temperature first. Before the temperature, carbon to nitrogen ratio. Carbon to nitrogen ratio very important. The carbon to nitrogen ratio we need to maintain at least 30 to 1. Carbon theater nitrogen or 25 to 1. If it is 25 to 1, it is more favorable. If it is 25 to 1. Give me a second. Okay, sorry. Okay, what we discussed, the carbon nitrogen ratio. Yes, for microorganism, methanospecious microorganism, we need to provide favorable environment. Therefore, their substrate should have 30 to 1 or 25 to 1 nitrogen percentage. Okay, carbon to nitrogen ratio. If it is dry leaf we use, at least if you think if you use 30 kilogram of straw, then we need to at least 1 kilogram of cow dung, like this, 1 kilogram of kitchen waste. Kitchen waste is a nitrogen source. Kitchen waste is a nitrogen source. Therefore, we need to add dry matters as well for better development of microorganism. And the second condition is they need temperature. They need temperature at least 35 Celsius. If it is more than 35 to 45, no problem. 35 to at least the minimum 35 they need. Otherwise, in cold condition, they can't do this process. 35 to 60 Celsius, okay. 35 to 60. Okay. Now the third one, the moisture condition. Moisture condition inside the production tank, it should be normally if we adding 5 kilogram of material, we need to add 5 liter of water. Remember, 50% of water. 50% of water we need to add to maintain the moisture condition. The other very important factor is the pH level of the mixture. The pH level we need to maintain at 7, close to 7, same as compost production. pH level maintain at 6.5 to 7 or close to 7. Close to 7. Close to 7. Close to neutral condition. Close to neutral condition. What else factors? Temperature, moisture, and how about oxygen level? Oxygen requirement for biogas production. For compost, how about oxygen percentage required for compost production? Last day I explained. How many percentage of oxygen required for compost production? Quickly. How many percentage of oxygen required for compost production? Can't remember? No, wrong. Timid wrong. Amar, what is the percentage? Amavi? Yes, very good, Tasha. No, Amar, wrong. Ashit, correct. Good, Adish. Amar, how we provide 25% in normal environment? 20%, no? 20.6 or 21%. Then if we need to provide 25%, we need to add additional supply එකක් දෙන්න. I'm asking not in biogas production, I'm asking in compost production. Last day explain. Compost production, what is the oxygen requirement? Amavi, what is oxygen requirement? Amar, think again. Go through the note again. Melfa? Shena, what is the oxygen percentage requirement for compost production? I told, I think you can remember I told in your home garden, if you make a pile, it take more than 3 months to make compost. But in if I provide favorable conditions, temperature, pH value, and moisture level, and carbon nitrogen ratio, we can produce this compost by at least 1 month. Okay, only one answers yet. For compost production, the oxygen percentage is 5%. 5%. It is very low level. They are a microphilic microorganism. The respiration microphilic microorganism we use. For biogas production, what do you think? How about the oxygen percentage required? Oxygen percentage required for biogas production? Very good. So it must be a 0%. 0%. If it is 1% of oxygen, it is a toxic condition for microorganism. It is toxic condition for microorganism. Therefore, we need to fully cover, fully sealed, closed environment provide for the microorganism. Okay, they are the main requirement for biogas production. First of all, we need to provide good substrate, carbon to nitrogen 30 to 25 to 1. Good substrate for microorganism. After that, we can introduce, same as dairy production, we can introduce microorganism by already make biogas unit, sludge amount, small amount of sludge we can add to our mixture, our substrate. Then we can introduce microorganism, methano microorganism for our biogas unit. Then we if we can maintain the conditions inside your system, you can make biogas and you can use as an energy source. Okay, that's a digester. The digester is a raw material is collected and microorganism act on this digester. Okay, that's a gas holder tank. And biogas out, we can get into this direction. The effluent, that's mean already digested particles, we can collect from this effluent tank, outlet tank. Okay, that's the biogas production unit. The biogas unit production process, we can divide. Conditions you can note down here. Biogas production condition, please note down here. It is not given in the tute. Okay, quickly note down. Not given in the tute. Biogas production condition. Quickly note down and let me know. Okay. Complete. Okay, now the biogas production process we can divide into four main steps. Four main steps. The first one is, I will write in the board, biogas production steps. Biogas production steps. Steps. Biogas production steps. Okay, the biogas production first step is the organic matter hydrolysis. It is very important. The organic matter hydrolysis. Okay, organic matter hydrolysis. You already know these words, I think. Hydrolysis. Organic matter hydrolysis. The second step is the fermentation. Fermentation. The third step is the acetic acid production. Acetic acid production. The final step, fourth step, is the methane production. As I told, methane production. As I told yesterday, the biogas is not only a methane mixture, but the main gas is the methane gas. The methane is a greenhouse gas, therefore it is actually, the methane is energy source, but if it is released to the environment, it is an environment hazard. It is not hazard. It increases the global warming in the earth. Okay, do you know what? You already know what's the meaning of this hydrolysis. Hydrolysis කියන්නේ මොකක්ද? What's the meaning of hydrolysis? You already know in the biomolecule lesson. Hydrolysis. Hydrolysis. What's the meaning of the hydrolysis? Yeah, good. Break down into simple, yes. Convert into simple form, yes. First of all, this organic matter, our substrate normally contain carbohydrate, protein, like complex structures. In first step, this organic matter convert into simple matters by hydrolysis. That we call as complex organic matters. Complex organic matter mean you already know carbohydrate, protein, cellulose, like complex compounds convert into glucose actually, and amino acid, simple compound, simple monomers compound. Okay, that is a compound. Okay, that we called as a hydrolysis. The method of microorganism, actually they are doing first process is hydrolysis. organic material breakdown. Okay, after that, this simple compound like glucose, amino acid, this simple compound in the fermentation process, simple compound react with microorganism and convert into acetic acid. Okay, just convert into acetic acid by microorganism activity. That we call as fermentation. By this simplest organic compound, then convert into acetic acid. In the production of acetic acid, carbon dioxide, hydrogen, ammonia, and organic acid. Not only acetic acid, it produces carbon dioxide additionally. Okay, carbon dioxide, that the process we called as acetic acid production. Okay, carbon dioxide, what else? Yeah, hydrogen, carbon dioxide, hydrogen, ammonia, NH3, and NH4 also. Different compound produced in the production process of acetic acid. The acetic acid is a main. Then in the acetic acid, finally by methanosphesis convert into methane gas. And the mainly produce methane gas, but it's not only composition methane. It's there are some other steps, other gases as well. Okay, the main gas is the methane in biogas production. More than 60% it is methane. Okay, you can note down. In the tube I have given space the steps of stage of biogas production, page number 31. You can note down. Okay, complete. They are the states of biogas production. Everything doing in this process doing by methanosphesis, anaerobic microorganism. I complete. Okay, now note down in the tute anywhere on a space the composition of the biogas. Always in MCQ questions asking the composition of the biogas. I'm going to erase the composition of the biogas. Composition of the biogas. I told mainly it is contain methane. Methane is CH4. Methane it's contain 55 to 65%. Average it is 60%. Average 60%. And next one is carbon dioxide. You already know carbon dioxide. It's contain approximately 30 to 35%. 30 to 35%. 35% carbon dioxide. The next one is hydrogen sulfide, H2S. Hydrogen sulfide. It is only 1%. Less than 1%. Okay, less than 1%. Less than 1%. The next one is nitrogen contain also less than 1%. Less than 1%. Hydrogen same, less than 1%. Less than 1%. And very small amount of carbon monoxide. Carbon dioxide CO2, carbon monoxide CO. It's also less than 1%. Very small amount. Okay, that is the composition of the biogas. Okay, any doubt in microorganism production? You can ask. Now the microorganism production part is fully completed. Every production is we completely covered microorganism productions. Microorganism production. In exam, there is a question in exam. They will ask deep question in biogas. No deep questions are not asking. They can ask what are the main steps of only one structure is coming. Then what are the main steps of biogas production? Then you should have ability to write down steps. What are the steps? The organic matter hydrolysis, fermentation, acetic acid production, and methane production. They are the steps. They can ask what is the composition, what are the main two gases in the biogas composition, methane and carbon dioxide. What are the main optimum condition for biogas production? Like no deep explaining questions, but point type questions can. Ashit, okay? Now everything we have discussed. Okay, good. Now the microorganism production is completed. Go to page number 32. Structure of plant, stem, leaves, roots, functional and economic values. this time this part actually no more questions coming, therefore I will rapidly complete this part. And the forest also same, our next part, forest also very small part, simple part, only 3 types of forest and their features and example you need to remember. In the tissue culture we can discuss what are the steps, and tissue culture is important because this time most possible probably structured question possible to come in tissue culture, because nowadays the tissue culture is most used in agriculture and in technological vegetative propagation. And you can read the final part that the invertebrates, vertebrates, I'm not going to discuss again. That's your O-level lesson. Invertebrates, vertebrates. The final part you can read and understand. No any spaces here. Full note I have given. Full note I have given. You can understand vertebrate, invertebrates, fish, birds. Okay, you can understand the first final part in tute, reading the tute. Okay, up to tissue culture I will discuss with you. Okay, do you need break? Still 8:30. We'll continue, no? Okay, plants. Okay, we're going to start about plants. Okay, plants. Go to the page number 22. Plants. Do you know plant kingdom is plante? Plante kingdom. We can, I think you already know the classification. That's a classification that I not given in the tute. This is the plant classification. Plant classification. Okay, this is a plant classification. I will show you. Okay, this is the plant classification. Wait. Okay, this is a plant classification. The plant kingdom, we can divide into non-vascular plant and vascular plant. Vascular plant and non-vascular plant. Okay, do you know what is the difference between non-vascular and vascular? I think you know. Everything is given in the board. Non-vascular and vascular. What's the difference? Anyone idea? What's the difference between non-vascular and vascular? Vascular and non-vascular. Okay, I have given here. Vascular mean plant, they have vascular tissues like xylem phloem. The non-vascular mean they don't have xylem phloem vascular tissues. Vascular tissues. Okay, vascular tissues, vascular plant, again 2 types, seedless plant and seed available plants. Seedless and seed available. Seed available plant, again naked seed and covered seed, different classification. Okay, you don't need to remember everything, but get some idea. Okay, seedless vascules again divide into 2. Again divide into 2, and non-vascules again 2 types. Okay, that's a full plant classification. I will send this one classification in the group, then you can go through. you can go through. Okay, next, go to the tune. Oh my God, where is the note? Give me 1 second. Listen. Oh, four. Okay. Now 70% of the lesson is completed. We have only small parts. Okay, this is the lesson. Okay, plant structures, stems and leaves. Okay, now plants we can divide into main two categories. Can you hear me? Yes. Plant main two categories. They are monocotyledons and dicotyledons according to the seed types. According to seed types. In the seed, if it has two cotyledons, that we called as dicotyledon. If it is only one cotyledon mean like they are the cotyledon. Okay, I'll show you in the video the cotyledon. One cotyledon that we called as monocotyledons. Monocot and dicot plants. Okay, I have given here this one now clear. This is monocotyledons and dicotyledons. Okay, monocotyledons and dicotyledons, their characteristics is given. You can refer the note. Monocotyledons, they have fibrous root system. The root system you already know in the O level, and taproot system for dicotyledon, like mango. For monocotyledon, like coconut. Stem usually does not branch, yes, coconut. Stem is well branched like mango. Mostly uniform in diameter, yes. Non-uniform in diameter. Parallel venation. Yeah, the leaf structure. And mainly floral parts, tree. In monocotyledon, it is tree. In dicotyledon, it is 4 to 5. Please go to the tute. Number of cotyledons, 2 in dicotyledons. Okay, they are the characteristics. Okay, I will show you what are the differences between monocotyledon and dicotyledons. This is the dicot plant leaf. Okay, dicot leaf. There, that's a cotyledon. These are the cotyledon. The different parts. This is monocotyledon. Dicotyledon. You can understand the difference in leaf structure. That's the difference in leaf structure. Parallel venation. Reticulate venation. I have given in the table. Okay, understand the difference. Monocot and dicot. They are the root system, taproot system, and fibrous root system. This is fibrous monocotyledon. Taproot system. This is a taproot. Weak stems and strong stem in dicotyledon. The flowers in monocot and dicot different. Petals only three in but dicotyledon it is 4 to 5. It has 5 sepals. Okay, now I think you can, if exam asks, you can write down 3 to 4 differences between monocot and dicot. That enough. Okay, this part actually no more questions coming. Okay, plant leaves. The main function of the plant leaf is photosynthetic. You already know. The mainly two type of plant leaves. The same monocotyledon plant leaves and dicotyledon plant leaves. Monocot plant leaves and dicot plant leaves. Write down monocot plant leaves and dicot plant leaves. 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 Page number 33. Okay, I have given now difference between monocot plant leaf and dicot plant leaves. It is important. Monocot plant leaves structurally the structure is not parallel in dicot. You can see in monocot it is parallel. Dumbbell shaped guard cells, bean shaped cells, not differentiate into and spongy sophil. That this much you don't need. Okay, that's actually this the deep level you need for biology students. You don't need. Okay, I have given next the parts of a cross section of the plant leaf. Cross section of a plant leaf. Okay, this is a cross section of plant leaves. This is cuticle and upper epidermis. bundle chain, and xylem and phloem. You can see, you can identify they are xylems and phloems inside the xylem. Spongy cells and different types of cells in the inside leaf. Actually, they are not coming. If they ask at least one part, identify at least one part, you can remember this. Cuticle main cell types enough. No, don't need everything. Cuticle, xylem, phloems, and spongy cells, upper epidermis. These layers you can remember. Okay, they are the different parts inside a plant cell, plant leaf. Okay, next I have given each part function of the cell. Each part function of the cells you can read contains this cutin layer mainly for protection. Cutin layer, the lower epidermis, this epidermis mainly two types, lower and upper. Lower epidermis important for reduce water loss, and contain uh, stomata and guard cells. Okay, you can read and remember if you can remember. Actually, not compulsory. This every functions, everything functions not compulsory. Okay, now I have given photosynthetic reaction. Photosynthesis reaction. Photosynthesis reaction. Okay, there is a mistake here. This is a mistake. This part. I'll draw it correct. I'll write the correct one. The main raw material for photosynthetic, you know, this equation, you know, CO2 plus H2O. Then it is produced C6H12O6 plus O2 plus energy. If it is balanced, if I balance this equation 6, 6 and 6. Okay, this is a photosynthesis equation. Photosynthesis equation. Photosynthesis equation, it has a mistake. And always asking these photosynthetic pigments. Photosynthesis pigments are chlorophyll and carotenoids. Chlorophylls and carotenoid. I think in one time there was a structured question: what are the two main types of photosynthesis pigments? Carotenoid and chlorophylls. Chlorophylls and carotenoid. There is another one that we call as xanthophyll. Xanthophyll, it is less cooperation for the photosynthesis, but that's also we can write xanthophyll. Xanthophyll. The carotenoid and chlorophyll are the main photosynthesis pigments. Gases exchange, you already know, carbon dioxide and oxygen. Then I have given economic importance of leaves. Economic importance for human food, animal food, fertilizer production, textile industry and medicine users. We can use this plant tissue, leaf. Plant leaf main function is photosynthesis, and other industrial uses as well. Industrial uses as well. Okay, plant stem. Plant stem also main two type: monocot stem and dicot stem. Monocot and dicot. Monocot and dicot. Okay, This one, you just need to identify difference between this monocot plant stem and dicot plant stem. Monocot and dicot. I have given here the difference between monocot and dicot, not differentiated into monocot, not differentiated into cortex and pith. See, in this is monocot, no differentiate cortex and pith, no specially designed separated. But if you go to dicot, it is separated, pith and cortex. Pith and cortex, it is separated. You can see clearly, you can identify, but it is difficult to identify. It is not well separated. Monocot plant stems mainly contain parenchyma tissues. Parenchyma tissues, and the dicot what happened? Monocot mainly parenchyma tissues, but in dicot it is parenchyma and collenchyma both tissues. Parenchyma and collenchyma both tissues. You can see in dicot it has a special arrangement in dicot, but in monocot it don't have special arrangement. It is like a randomly arranged. This is important. In monocots, no any secondary growth. After matured, it is no any, after that no any maturation. After that no any growth. But secondary growth mean after actually if I cut the stem, again no growth. Okay, matured වෙලා මේක කැපුවොත් ආයි හැදෙන්නේ නැහැ. But in dicot allow secondary growth. Varying in size, but in dicot uniform in size. Okay, they are the parts and functions of each parts. I have given they are the comparison dicot and monocot. You can make a short note. These bundles are separated, but it is not well separated in monocot. That's the main difference. Okay, then I have given each part of the function. You can read and complete, make a short note before next day. Okay, now I'm going to more into the each and every part. The secondary growth I told, scalar cambium. Okay, I'm going to root. That is root. Plant root. Okay, go to plant roots. page number. Okay, this is the plant root. Plant root structure is missing. Epidermis cortex. So okay. Oh, sorry, it is the dicot and monocot root figures missing in the tute. I'll send the picture into group. Dicot and monocot root structure. Okay, that is a difference. Please make a short note of this part. This part is not too important, but you can make a small short note with parts and there are differences. Then it is easy, I think. Okay, without taking time, I'm going to discuss about forest, and finally, today we need to discuss about tissue cultures as well. Then if we can discuss complete the tissue culture, now next week we have this cell structure we can complete today. The final part also you can make a short note. The economically important vertebrates and invertebrates. Then go to discuss about forest and tissue culture. Okay, forest. Forest mainly, if I discussed last day also, forest. Forest how we use for the technologies. Actually, that we discussed. The forest mainly two types. Forest mainly two types. According to the population of the plant. Forest can be divided into two types: primary forest and secondary forest. The primary forest mean actually they are biodiversity, and it is actually natural environment. That's a primary forest, not influenced by man. Okay, that's a primary forest. The bio that's this type environment. The biodiversity is very high. Biodiversity is rich in biodiversity, and there are different types of plants and different types of age of these plants, like a forest, like a sinharaj, like a kanni. They are primary forest. The secondary forest is influenced by man. It is the vegetated by man, and biodiversity is low, and it is their plant size and plant age are same. The uniform cultivations, they are the secondary forest. No more large plants, they have only small uniform plant population. Okay, types of forest. Okay, in this lessons, no essay or structured essay, only at least one MCQ. Therefore, we don't need to take more time. That's why I'm doing rapidly. Forest mainly three types: tropical west forest and dry mixed evergreen forest and hill country wet zone forest. Okay, I told last day what are the examples for tropical wet zone forest: Sinharaja, Kanneliya, Dediyagala, Nakayadiniya, Morapitiya, Bomburu, Botu, Bambara Botu. Sorry, Bambara. They are the main trees or large trees, no, and they are height between of trees 30 to 40 meters. The height is very comparatively very high with other forest. See, this type forest you already know. Tree, several layers, that's important. Several layers, you can see the ground layer, and in here I have given canopy layer. They are the tropical wet zone forest. They have typical layers we can identify in different heights. Okay, you can remember these layers. Color of the trees dark green. Yes, it is dark green. The canopy formed at the top of the there is no light inside the forest. The dark environment, because the large trees make as a canopy, like a cover. Okay, therefore no more lights come to the ground layers, like a dark environment. Plant growth density is very high. Yes, undergrowth is the undergrowth is very low due to the low light conditions. High biodiversity is comparatively very high with other forests. They are the popular plants and most available plants in the tropical wet forest, wet evergreen forest, like Sinharaja, Kanneliya, you already know. Okay, simple. They are the canopy layers, emergent layer, canopy layer, understory layer, forest floor layer, understory layer, canopy layer, and emergent layer. Okay, there are four layers. Plants grow with layer by layer and can like a canopy. Then the in low floor layer light condition is very low, the dark environment. Therefore, the undergrowth growth development is very low, not like other forests. Okay, come to dry mixed evergreen forest. Dry mixed evergreen forest. Last day we already discussed. They are Somavatiya, Vilpattu. You can remember Vilpattu is a dry mixed evergreen forest. Maduroya, Udawalava, Vasgamu, Yala, and Mahavali areas. Features: height of trees in wet evergreen wet forest, it is 30 to 40. Now it is 20 to 30. It is less. Trees growth during rainy seasons, growth rate is less during the dry season. You know, it is in dry zone. There are two seasons: dry zone and rainy zone, rainy time. In rainy time, the growth is high. In during the dry time, the growth is low. Bushes of 2 to 3 meter in height can be seen. The forests are light green. This is color light green, and hardwood trees. If you go to types of trees, you can see they are hardwood trees. Lot of stem. The valuability of stem is very high. They are in dry mixed evergreen. Veer, mill, palu like plants, important plants. Okay, hill country, wet zone forest, that you can easy to remember examples. Samanalakanda, Pidurutaragal, they are all a mountain. Horton Pains, Knuckles, and they are the features. Average height 8 to 10. 8 to 10. You can remember in evergreen wet forest, what is the average height? 30 to 40. In dry, the second one, evergreen dry mixed one, it is 20 to 30. This one at least maximum 10. Very small plants. With the increase in the it should be latitude, not that altitude. Yeah, altitude. Height of trees get reduced gradually. This grows as a canopy. You can see as a canopy. They grow as a canopy. Small plants. Small plants. And they are the importance of forest. They are the importance of forest. You can read and understand, maintain the atmospheric oxygen carbon dioxide balancing. For natural cycling, this forest very important. Protection water catchment areas, yes. In rain forest, mainly important for maintain the water balance, to circulate the water cycle. This is very important. This is very important. Prevent soil erosion, yes. To maintain the soil fertility, yes. Provide protection to pollinators and seed and fruit. Actually this biodiversity. For biodiversity in the world and in our country, these forests are very important for the actually for the tourism. For the tourism, for get additional foreign income to our country, this forest very important. And sustained tourism I have given. Natural beauty, as a natural beauty, provide habitat for living things, yes. Windbreakers, yes. Windbreakers, or sometimes the plants act as wind barriers, therefore they are windbreakers. Protection of endemic species, yes. Endemic, that's mean very reducing the population animals and plants, we can protect inside the forest. Okay, endemic organism we can protect inside the forest. And education and research purposes also, they very important. Okay, this is forest and plant. Now I am going to start tissue culture full lesson. If we complete the tissue culture today, that enough for today. Do you need break? Tissue culture, we need to discuss every process of productions. Do you need break? Yeah, we'll take a break. And now 9:20, we start 9:40. You have 20 minutes, then we need more 30 or 45 minutes for complete the tissue culture, then the cell structure is completed. Okay, join 9:40. I'm starting 9:40 and I need to complete quickly as soon as possible. Okay. Tissue culture. Okay. How we use tissue culture and what are the technologies we used in the tissue culture. Okay. First of all, you need to understand what is the meaning of this tissue culture. Now you know different types of every types of tissues we discuss: meristematic tissues, permanent tissues, then simple permanent tissues, complex permanent tissues, everything, the tissue type we discussed. Now the tissue culture, how we define this tissue culture? Actually, you should need to know a proper definition for the tissue culture. A tissue culture is a techniques grow a full plant by a cell or whatever collection of cell, whatever tissue actually. Simply by a small part of plant, make us a whole plant, make a whole plant that we called as a tissue culture. The tissue culture refers technique growing cells, tissues, organs in a nutrition medium under sterile, otherwise aseptic condition. Aseptic mean no any microorganism. If there are any at least one microorganism enter this system, all population will destroy. Okay, therefore this is a very critical process. The aseptic condition is must. Aseptic condition must. Okay, the tissue culture is a technique that we can use whatever plant part grow in a nutrition medium under aseptic condition, then we can convert this as a whole plant that we called as a tissue culture. From any small part, think example leaf part එකකින් whole plant එකක්, not only one plant, the large population of whole plant taken from the small plant, mother plant part that we called as the tissue culture. I will explain how to make, what are the techniques we need to follow to get this uniform population. That's a very important thing. We can make a uniform population. The characteristic are unique to each other. They are uniform. No any more difference between plants. No more difference like seedling plants. Seedling plant, you already know that's sexual propagation. That's a vegetative propagation. In sexual propagation, you know, one plant is totally different from other plants in the population. The uniformity is low. But in vegetative propagation, vegetative mainly in tissue culture, there are uniformity. The population, the daughter population, the uniformity is very high. The uniform actually, the production plants are uniform. Therefore, when it is adapt to environment, when it is placed in the field, it is management is very easy, because all vegetation is in the same field, it is same age, and their characteristics are same. Therefore nutrition requirements and light requirements and other all requirements are same for all plantations. Okay, therefore their management is very simple. Okay, they simply understand the tissue culture is a process. One plant part, think it is leaf plant, small part of this plant that we called as explant that we called as explant. This small plant part grow on a nutrition medium. Think it is a nutrition medium Under aseptic condition, mean without any microorganism, the maintain the aseptic condition is very important. Okay, then from explant, any part of the mother plant, we grow on a nutrition medium under aseptic condition and the favorable climate condition. Climate condition is very important for this tissue culture. I will explain what are the favorable climates and environmental requirements that need. Okay, this method also called as micropropagation. Yes, the micropropagation is a one method of tissue culture. Okay, now do you know what is the meaning of this totipotency? Totipotency, what is this? The totipotency ability of plants. Totipotency ability. Totipotency ability. What's the meaning of the totipotency ability? Anyone? Don't know? Totipotency ability. What's the meaning? It is the for tissue culture. That's the main keyword for tissue culture. If it think plant don't have this ability, then we can't do tissue culture. If plant, actually every plant has this ability, the totipotency ability, that's why we can do this tissue culture process. Yes, very good. The totipotency is the, think one cell of any plant, one cell, think it is a one cell has all information developed as a whole plant. මෙයාගේ තියෙන all information it contain developed as a complete whole plant. One cell is enough. Okay, single cell contain all information to plant develop as a complete whole plant, that we called as a totipotency ability of the plant. Okay, the plant has this ability, therefore the tissue culture is small part of the plant. We make a whole plant population in a nutrition medium under aseptic condition in a controlled environment condition. Aseptic and controlled environment condition, growing any small plant part in a nutrition medium. Okay, in tissue culture, the small plant part we selecting, that we called as explant. In tissue culture, the callus mean, think, these words are very important. The totipotency ability mean one cell contain all information to develop as a whole plant. Okay, that's the totipotency ability. Plant has this ability, therefore we can do tissue culture for plants. Okay, tissue culture for plants. The second one is explant. This word is very important in tissue culture. Explant is what? The selecting plant part for the tissue culture. Actually, the mother plant part. Okay, that we called as explant. The other thing, other important word is callus. Callus. Okay, the callus I will show you. Okay, that's a callus. Actually, that is a tissue culture. A small plant part grow in a small bottle inside a small jam bottle. Actually, normally we use jam bottle size bottle. Okay, that's a nutrition medium. Nutrition medium. When we grow under aseptic condition inside the nutrition medium, then you can see cell dividing and make as a cell collection. Cell collection. See, an undifferentiated mass of plant cell that form as an on a nutrition medium that we called as a callus. That's a callus. Okay, they are the callus. This is a callus. This is a callus. You can see undifferentiated cell collection. Okay, that's the already planted one, explanted. Okay, they are the callus. They are callus. The cell collection. They are callus.
I know it's been a while since I posted a video, but I just wanted to come in with some cool, exciting stuff. So right here, we're going to go ahead and take a look at some embryogenic callus.
Okay, they are the callus.
What to expect when you have embryogenic callus, and what to do to make you get more pups, pretty much. So here we have below for us a seed that was started on embryogenic media, so a certain cactus media that I use that has hormone levels that pretty much make it expand. I did this on a seed, so the tissue that first emerged was embryogenic seed tissue, and I've slowly been subculturing it onto other media to get regeneration.
Okay, now you have idea what's a callus. The callus mean, think it is a small plant part, small leaf plant, then that we can grow on a small bottle in a nutrition medium that we can propagate. Okay, propagate in a aseptic condition, and we need to seal this bottle and maintain the environment condition. When it is control the environment condition, then the callus formation like a undifferentiated cell collection like this. The plant part, plant leaf එක මේ විදිහට grow වෙනවා. Then this plant callus that we called as a callus. මේක තමයි callus කියන්නේ. That we called as callus. This callus, again, we can divide into small parts. Again, we can cut into small parts. Again, we can cut into small parts and we can add into another bottles. Make a මෙහෙම bottle 10කට වගේ දාගන්න පුළුවන්. Small part is enough. Only few cells, at least one is enough actually. එකක් තිබ්බත් ඇති for growth. Okay, that we called as a tissue culture. Now you can see we can make a large population, more than 1,000 large population by a small part of the mother plant. Okay, that we called as a tissue culture. Okay, I think now you have idea what's the meaning of the tissue culture. Okay, now I have given essential requirement for plant tissue culture. Essential requirement. Okay, the explant, I think you know now what's the meaning of explant. The plant part. the plant part is the explant. The next one, a nutrition medium. Yes, the nutrition medium inside the bottle, a proper nutrition medium is compulsory. Okay, to growing. When change the stage of plant growing, we need to maintain the nutrition level. I will explain how to change that. When it is shoot development, root development, hormone requirement is it is changing. Therefore, we need to manage well. Okay, we need to manage well. Then third one is growth regulators. That's mean hormones. Growth regulators mean hormones. There are three main hormones we use in tissue culture. Remember, there are three main hormones: auxin, cytokinin, and gibberellin. Auxin, cytokinin, and gibberellin we use as plant regulators. Plant regulators, and it need carbon source. In tissue culture, as the carbon source, mainly we use charcoal. 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 Mainly we add in charcoal. And amino acid, vitamins, and antibiotics required. And solidifying agent that we use, agar. Agar. Agar tamadan. Like gelatin, solidifying agent. Proper physical environment, temperature, yes, temperature, lighting condition, humidity, pH value, oxygen, and the lighting period. Photo period mean lighting period. All we need to manage for the favorable for the plant development. According to the stage, we need to manage all these environment factors. And strict aseptic condition, it is very, very important. This aseptic condition, if it is, I told when it is planted, think planted million of plants from one mother plant, millions in the population of vegetation, then it is a huge investment. The initial cost is very high. If at least one microorganism enter to this system, the entire millions of plant is destroyed. That's the risk of this plant tissue culture. එක microorganism කෙනෙක් ගියොත් මේ million එම lost වෙනවා. Okay, that's the million එම destroy වෙනවා. That's the risk of this laminar flow cabinet. I will show you. May to keep the aseptic condition, every process in tissue culture, this explant preparation, explant එක කොහොමද ගන්නෙ, ඊට පස්සේ explant enter to the nutrition medium, everything we doing in a laminar flow hood or laminar flow cabinet කියලා කියනවා. Laminar flow cabinet. Laminar flow cabinet. Okay, I'll show you laminar flow cabinet. It is maintained, it's like a cabinet. It's maintained the aseptic condition. It's maintained the aseptic condition. Okay, that's a laminar flow cabinet. Okay, you can see one person is doing tissue culture in the laminar flow cabinet. Okay, that's a cabinet. It is just cabinet, like the shape is cabinet. Okay, this one, it is covered, fully covered by glass, and it has a air sucking machine, and entire the inside air already available. Wait, I'll show you. Already available air is filtering and again provide to the system. Okay, I'll show you it.
Backed by the overhead system, having a high efficiency particulate aggregate, that is HEPA filter. It is used to maintain asepsis while
Yeah, HEPA filters mean HEPA filters mean it is a special type of filter that is filter out and come air inside. Okay, no any microorganism coming into the system.
preparing medications and parenteral nutrition. Use laminar flow after cleaning and dusting the room. Clean the glass surface with damp cloth soaked in soap solution.
Okay, that's how we make a sterile condition.
Steel surface inside the laminar
Normally
flow
for
system.
this cleaning process we use 90% alcohol. It is more toxic for microbes.
In each shift.
We need to
Turn
turn
on the air
on
supply at least 30 minutes before
30 minutes
use.
keep for
Keep the
sterilization.
front door open during this time so that the non-sterile air can be replaced by clean sterile air. Close the front panel after 30 minutes and leave the blue light on for a period of 30 minutes. Switch off the blue UV light and open the front panel. Switch on airflow so as to maintain pressure inside the laminar system at 8 to 12 centimeter above the atmospheric pressure. Notice this by the movement of the red water column on top of the instrument. Switch on the tube light. Laminar flow is now ready for use. Ensure strict asepsis by wearing mask, sterile gown, and gloves while preparing the medication. Switch off the white light and airflow after preparation of medication. Cultures to assure the quality of HEPA filter. Switch off the fans and doors should be closed by physical transport.
Okay, they are the HEPA filters mean the filters, special filter that we use for the filtering of outside air when it is coming to the laminar flow. Okay. Okay, they are the requirement for plant tissue culture. They are the main requirement. We need nutrition rich medium. Required nutrition level include nutrition medium, growth regulators, carbon source that we use charcoal, amino acid, vitamin and antibiotics, solidifying agent that we use agar, environmental condition and aseptic condition. To maintain the aseptic condition, we use laminar flow cabinet. Okay, now we have all requirement. I'm going to make a explant now. Preparation of explant. Okay, explant preparation, any plant part. Okay, I have given in here leaf pieces, shoot tip, root tip, young stem, inflorescence, flower bud, fruit part, embryo, pollen grain, whatever part we can use for the tissue culture. Okay, whatever part is possible to use. That is the explant. Explant preparation. The explant should be collected from the healthy and free from disease. Okay, it should be healthy and free from disease. If it contains some disease, microorganism enter the production process through the explant. Therefore, we have to careful. We have to careful, and it should come from young and actively growing plants. Yes, yes. This tissue should be easy to sterile. It should have good potential to grow and regenerate new plants. The explant size should be it is appropriate to the nutrition medium. Okay, think the explant is this size, the nutrition medium is this size, then we cannot enter that. Okay, therefore that should be appropriate to the size of the explant and nutrition medium. Okay, then any plant part we can use. You can remember few examples. Okay, now the explant is okay. The explant mean any plant part. Okay, now I need nutrition media. Nutrition media. Okay, now the nutrition medium used for the nutrition medium, it should have all macro micronutrition for the plant cell growth. Okay, I have given in here macronutrients, micronutrients, vitamin, carbon source, growth regulators, and activated charcoals. Charcoal also we need. Activated charcoal and carbon source, vitamin, micro macronutrients I have given in PK and other micronutrients. Okay, that's a balanced nutrition medium we need for the tissue culture. And in commercially we use MS medium, and there are different names, and here I have given White's medium, different commercial medium as well that we can use. If not, we can make by adding ingredients, we can make our own nutrition medium. Okay, any method. However, we need balance and required level nutrition medium. Okay, now if you have, now what we have laminar flow cabinet, that means proper environment, and we have explant, and we have nutrition medium. Okay, now I want to place the new explant inside the nutrition media. Okay, inside nutrition media, and in aseptic condition in the laminar flow cabinet, and we need to maintain the after place the explant on the nutrition media, we need to maintain the requirement for callus formation. Oxygen, more oxygen is required. Callus formation, oxygen, not oxygen, oxine hormone should be high concentration. For shoot formation, callus form, remember after callus formation, we again this callus cut into pieces. Yeah, understand that? For this callus formation, the first tube, it need more oxygen. This nutrition medium, more oxygen is required. But after that, they introduced to new medias. The new media make shoot and roots. For that, they need more cytokinin percentage, more cytokinin than oxygen. Okay, therefore we need to always manage this nutrition medium condition for according to the plant growth condition. Okay, and after make the nutrition media and everything okay, now we need to maintain the tissue culture lab. Okay, aseptic condition maintain window, culture room with controlled temperature and lighting condition. Okay, distill water unit, pH meter, pH management, micropipette. There are different the climate and environment condition we need to maintain around the plant now. Okay, when it is maintained, when it is maintained, okay, when it is maintained after required growing time, we can make a tissue culture plant. Okay, I will show you the final tissue culture plant. Okay, they are the tissue culture plants. You can see inside the bottle it is, they are the, it is, now they are the, without open, they well grown. Well grown, they are the tissue culture plants. Tissue culture plant. Okay, now, can we directly introduce this plant, tissue culture plant, into the environment? Is it okay? Is it, can we directly introduce them? I asked, this plant, can we directly grow in the field? No, we can't, because they are they still growing in the controlled environment. Their strength is very low, and they are very soft, still very soft. The මේවා නිකම් මෙහෙම ඇල්ලුවත් කැඩලා යන දේවල් එක very soft part tissues they have. Therefore, we need to adapt this plant into outside environment. Okay, for this adaptation period, we need to gradually increase their lighting condition, temperature condition. Gradually, we need to practice this plant into outside environmental conditions. Then we can introduce gradually into the field, that is we called as the tissue culture. That we called as a tissue culture. Lot of questions coming on tissue culture. You can read, yeah, everything I have given. They are the steps: selection of the explant, I told, surface sterilization, I show you the using ethanol normally we can use. Then medium preparation, explant inoculate mean enter the explant into a nutrition medium. Inoculate කියලා කියන්නේ enter. Then callus induction, root induction, the final step, adapt to the finally normal environment, external environment, gradually adapt to external environment. Okay. Okay, that don't need, you can read. Application of tissue culture. Okay, that's important. Application of tissue culture. That is provide disease-free plant, free disease plants, and we can converse endangered plant by using tissue culture, because only one tissue is enough. And secondary metabolites, you already know, production of secondary metabolites and alkaloid and flavonoid. Okay, for this process we can use tissue culture. And we the other thing is, as I explained earlier, the uniform population we can maintain in the field. Therefore, the management is more easy. Okay, management and other practices very easy. Okay, that's all about tissue culture. Then the cell structure is completed. I'm not going to explain again invertebrates, vertebrates, that you can read, and you have full note in the tube. You can read and make a short note and go through that. Okay, then the cell structure is completed. Next week we start the electricity. electricity, and you have very limited time now. In our side, we have only cell structure complete, no, electricity and natural environment chemistry, environment chemistry also you all have, I think, full note. Okay, if anyone don't have, please, yeah, now all have. Today I posted for all. Okay, environment chemistry all have tudes, you can read, and the environment chemistry also don't have spaces you can read, only few spaces for this oxygen carbon cycles. Okay, others are given here. You can read and complete your works. After electronics completing, we come to this lesson, and rapidly I will explain the environment chemistry as well. Environment chemistry actually very simple. No more chemistry, the different environment effect from the chemical reaction. They are discussed in the lesson. Okay, that's all about cell structure. Now we again meet with tomorrow practical session, and now you have very limited time, approximate you have 8 weeks and only 10 weeks more. No, vertebrates and invertebrates, do I need to cover it? I thought it can make a short note. Mammals, birds, එක ලේසි නේද? මම කරන්න ඕනේද? Yeah, no any spaces in the tube. You can go through, I think. Okay, you can go through. If there are any doubt in the vertebrate invertebrate part, please ask. Amar, is it okay? I think it is in the class that we don't need to take time for this type one. Amar, okay? Okay, if we have time, I will explain by 20 to 30 minutes. Within 20 to 30 minutes, beekeeping. Okay, beekeeping I will explain. Yeah, before that, make a short note. Full cell structure lesson, make a small short note. Full cell structure lesson. Now you have full lesson. Now you can read the environment chemistry and get some idea. I will explain full environment chemistry, and next week we start electricity. And you have very limited time. You have, I think, the time is less than 10 weeks now. You have only. Therefore, do your maximum this few days, because your every entire life is decided according to this few days, your works. Therefore, don't take any break. Now I know this is the most difficult time I know to work, because very limited time now. This time is little difficult, I know. Therefore, somehow however you have to do work now, because this is the most important period to work. කලින් කොච්චර වැඩ කරත්, this is the most important time. මේ වෙලාවේ වැඩ කරේ නැත්නම් it is a problem. Okay, this is the most important critical time of your life. Actually, you have at least how many days for exam? November 10. Today's, Yeah, I think approximate 80 to 85 days. 80 days. If you're doing well, if you're doing your maximum, you can totally change your result within this 80 days. 80 means more than 2 months. More than 2 months, you can totally change your result. But you have to manage your time well, and you should have a proper plan. I can't give you a plan. You should have a proper plan to how you manage this within this the remain few days, how you manage this. Okay, you can work on every lessons, try to work based on the exam, whatever mock exams. In our class, we doing next week also you have exam. Next week, the exam number 5, structured essay and essay. Please ready. Next Monday, it is 3 hours, 3 hours, and it is a full paper, 4 structured essay and 4 structured essay and 6 essay questions. You can select as exam and answer. For essay part, you have selection questions. Okay, any doubt? Any other doubt? Any other question? Yes, I'll do bee and fish part. Yes. Correct. Revision class we can take arrange a time, no? 30 minutes I will take from revision class and I will explain. Next Tuesday we start the electricity. Any other question? No. Okay, then goodbye and good night. Work very well. Manage your time very well. That's important.